Hello, Assalamu alaikum. In this video, we will understand about the Krebs cycle. So, Krebs cycle is very important for production of NADH and FADH2 and ATP, but mostly FADH and NADH, FADH2 and NADH, which that is the precursor of ATP production, and the ATP is our energy currency. So here is the cell contain cytoplasm and the plasma membrane and nucleus. So this is the mitochondria contain inner and outer mitochondrial matrix. This inner mitochondrial matrix contain Krebs cycle. The function of the Krebs cycle occur in the inner mitochondrial matrix. This is the inner and outer mitochondrial membrane, which that is the bigger picture. Outer mitochondrial membrane contain MPC. The MPC is the MP channel. So in this way, the cytoplasm, the glucose when rise, the glycolysis will occur to produce ATP and NADH, which that is very less, not our need. So it is very less. So our need to more produce of ATP. So this pyruvate will be form 3 carbon containing 2 molecule of pyruvate. Further, if the oxygen level will be low, hypoxia will occur, and hypoxemia will lead to lactic acid fermentation will occur to produce more ATP. Because the oxygen is not present and the mitochondria required oxygen for production of the ATP as a aerobic respiration. The MPC channel allow to influx of the pyruvate, and the pyruvate will enter the 3 carbon containing pyruvate. So in this way, this pyruvate, when the NAD plus use and pyruvate dehydrogenase enzyme used to remove 1 carbon in the form of carbon dioxide, which that exhale during the breathing, and NADH will be formed. This acetyl-CoA will, under the action of hydrolysis, the Co and CoA will be separate due to the hydrolysis, and in this way the oxaloacetate will be formed. This oxaloacetate form from the malate. So remember the malate. So anyhow, this is the major mechanisms you should need to understand. So in this way, I hope you make sense about that. So in this way, basically that under the action of citrate dehydrogenase enzyme by the mechanism of condensation reaction to produce citrate. So this citrate is basically, that is why it is also known the citric acid cycle, because again and again the formation of citrate is known as the citric acid cycle. The citrate basically, which that is when remove the water in the presence of aconitase enzyme by the action of dehydration lead to produce cis-aconitate. Cis-aconitate is basically contain basically 1 2 3 4 5 6 carbon. So this 6 carbon containing molecule under the action of again aconitase enzyme and the hydration will lead to produce the isocitrate. This isocitrate basically contain basically is the 6 carbon, and in this way again NAD plus used for the isocitrate dehydrogenase enzyme to oxidative decarboxylation to produce again the 1 2 3 4 5 carbon, and the 1 carbon dioxide also remove. The alpha ketoglutarate basically contain here, and again carbon dioxide when remove. So in this way the NADH will be formed again due to the alpha ketoglutarate dehydrogenase enzyme to remove hydrogen, the proton. And in this way, this is the oxidative decarboxylation will occur. In this way, the product will be succinyl-CoA, because the addition of coenzyme, this coenzyme basically add, and the hydrogen will donate to the NAD plus to the NADH formation. The succinyl-CoA, with the succinyl-CoA synthase enzyme used to succinyl-CoA synthase enzyme used to phosphorylation. So the GDP will convert into the GTP, and in this way the GTP will convert into the GDP again to produce ATP. But anyhow, this is the succinate formation will occur. The succinate is basically the 4 carbon containing, and the carbon dioxide further remove in the succinyl-CoA synthase enzyme during during the phosphorylation, so that carbon dioxide will be exhaled. In this way, you can see the removing of the carbon dioxide will remove and decrease the carbon number to form a succinate, which that's contain a 1, 2, 3, 4 carbon. And anyhow, in this way, FAD plus will use for the succinate dehydrogenase enzyme for dehydrogenation to produce FADH2 and to produce fumarate. And this fumarate is basically contain again 4 carbon, but that you can check out that the double bond will be formed by the removing of the proton again. In this, the addition of the water basically will lead to fumarase enzyme to hydration to produce malate, and that malate again used for the production of oxaloacetate by the dehydrogenation of the malate dehydrogenase enzyme. So in this way, I hope it makes sense about that. In this way, the malate, which that is the 4 carbon containing, will become again a 6 carbon containing molecule by the addition of the acetyl-CoA by the removing and the hydration. So the malate dehydrogenase enzyme used to produce NADH again, that's dehydrogenation mechanism to produce oxaloacetate. Oxaloacetate accepts the acetyl molecule. The basically the pyruvate will fix by the acetyl-CoA, lead to produce again the citric acid, the citrate. And anyhow, this is the Krebs cycle, and this is also known as the citric acid cycle. So this basically producing the NADH, FADH2, and the little amount, even one cycle produce 1 ATP. So neglect the ATP, but consider the NADH and FADH2. So in this way, here you can see the pyruvate dehydrogenase enzyme used to produce the acetyl-CoA, and that fix into the oxaloacetate. So anyhow, I hope you make sense about that. This is the Kreb cycle, a detailed video, which that is short, but you can understand as well. So in this way, the carbohydrate digest in your digestive system to produce glucose simple when enter into your blood. This entry into your blood will consume as a glycolysis and the Kreb cycle to electron transport chain. Thanks for watching. Bye.